Right now, it's my pleasure to introduce our last speaker from the HQP, Kyle Dews, a PhD student at the University of Alberta under the supervision of Dr. Brad Pino. Our Theme 6 lead, Kyle's work focuses on silviculture and stand dynamics, exploring how forest management practices influence structure, function, and resilience. And I hope your talk wasn't changed for the title, but Boreal mixtures, associations with below ground root prevalence. So let's welcome Kyle to the stage. Go ahead. I don't want to trip.
Thanks. Thank you. So as Kevin said, my name's Kyle Dews, a PhD student at the University of Alberta. And I'll be talking about kind of some of the below ground stuff that's happening in our boreal forests. So if anyone knows me, I like to start my presentations off by taking a hard left turn. So can I get you all to stand up? Yeah, like seriously, yeah. Have your seat. I know we're all kind of fading as we get into like the later part of the afternoon. So what I want you to do is do some like arm swings, like cross-body arm swings, and I'll tell you why. Yeah. So I'm waking you up because I know I saw a couple of you falling asleep in the back. Look at you. So swinging your arms, what it does is it'll basically get the left side of your brain, your right side of your brain talking to each other. So we're getting the creative and the analytical side working together, and everybody's up again. Okay, now you can sit down. Good. Now that everyone's kind of paying attention again, I recently had the opportunity to fly through the Atlanta airport. And when I was flying through it, I saw this display in the international terminal. For anyone who doesn't know, this is an art piece called Veneers. And what this artist did is they took sections, tree cores, from 15 different species, painted them, and turned them into these giant glass panels. And as I was sitting there thinking about this, is all I could think about is trees are everywhere. Is we as foresters, researchers, often get siloed into thinking about the impacts of our work. Is that one day we might be studying dendrochronology and tree cores, and the other day somebody's turning it into artwork. So I just like to kind of start off with that fun fact, and as we move in. So this study, to kind of hit you with the words first, it's a study happening up in Peace River, Alberta, so kind of that transition area that we talked about a couple times previously today. And the main study species that we're looking at here are trembling aspen, lodgepole pine, and tamarack. So for any of you student listeners out there, that's a true conifer, a deciduous conifer, and a true deciduous tree. So it kind of covers the gamut of potentials that will be seen in the boreal forest. And then inside of each one of those three species, there's crosses and singles of each species at two different spacings, so 25 centimeter and 50 centimeter. For you don't know, that's like this. And then they're planted in 3 by 3 meter squares, so that's like 200 trees, those ultra high density groupings. And the reason we're doing this is to play with mini rhizotron tubes. How many of you are familiar with mini rhizotron tubes? Okay, that's more hands than I thought it was going to be. So mini rhizotron tubes, my lab mate Andy here, he's kind of Vanna Whiting it for us, are these hollow glass tubes. These hollow glass tubes are put in the ground, and then trees are planted on top of them. So what it allows us to do is to look at what's happening below ground and above ground. So we can kind of tweak out or tweeze out some of what's happening below ground. So looking at things like root quantities, lengths, diameter. It kind of goes back to the last talk of talking in situ. This is in situ root measurements. So anyways, we planted these plots about 2 years ago now, and they're currently growing with, again, as we said, mixes of each one of those species. And what we're trying to see here is what is happening below ground and above ground and seeing if those kind of echo out, right? So if we're seeing trends in increased amounts of root prevalence, are we seeing expression of traits? Because something like lodgepole pine might have a taproot, whereas aspen might have more of a vertical or more of more of a horizontal spread. We're trying to tweak those things out of like what's actually happening here. So this is just kind of a diagram to show you what these root tubes look like. Is that they're just planted diagonally in the ground, and this allows us to test and look at things relating to both depth, and time. So it's like how long does it take a tree to reach certain depths, and then what are the percentage of roots covering those tubes are kind of what we're looking at, as well as we can measure variables like root car diameter and height of those individual trees. So how many of you have ever looked at tree cores? Okay, so you all, a good percent of you know like the pain that it is to like painstakingly sit there in the software and look at this. Roots are about 300 times worse than that. You're in this software and you're going and you're measuring little bubbles of every single root. So we're meticulously going through and taking images as you move down the tube. And then we're going through in the software and we're measuring the length of these, the diameters, here, I'll just go to the next screen to kind of show you what variables they are. So we're looking at counts, we're looking at lengths, volumes, area, and then we can average those out and compare them between treatments and spacings, essentially. So this is what they look like in situ. So looking at from 1 year to the next is that you can see in these two images. So this is a monoculture of aspen planted at 25 centimeters, so 196 little aspens growing above these two tubes. Or sorry, this is actually just one tube at 2 years. So this is 4 images because how the scanner works, it takes 4 images as it moves down the tube. And you can see the two tubes there, or sorry, the single tube at 2 different years. So this is 1 year post growing and then year 2 post growing is what you're seeing right there. And so kind of moving into the preliminary results is there are none. Surprise. Um, what we're doing right now is we're really still in this measurement phase. This is the first project of my PhD, and I think the bane of everyone who works in forestry is that trees take a long time to grow. So we're sitting here currently, we're measuring. We've got the preliminary year 1 to year 2. And where we're sitting at with those is that the year 1, the field we put it in is an old farm field, and it was tilled directly before we started planting on it. So there's a lot of organic matter gaps and other stuff in our year 1 measurements. So we're most likely going to have to exclude that. And so I didn't want to present on and kind of give you things that might be confirmatory. And essentially what we're doing now is just letting these trees grow and measuring them. So that's everything I was trying to keep under my 15 minutes, which I think I did with time for questions. So thank you all for listening, and please hit me with questions.
I wasn't even ready for that, Kyle. Thanks.
Yeah, sorry, I was trying to keep to my 15 minutes, so I think we
You're
have
quick.
5 minutes for questions.
Yeah, any questions? Harry.
Sorry, Kyle, just because I'm not a forester.
Yeah.
The reason you're expecting with the different densities is you expect to see a change? And just tell me what your hypotheses might be.
Yeah.
Yeah.
So when most of these experiments, so there have been experiments similar to this done, but they're generally done at wider spacings and over longer periods of time. So what we're trying to see is, well, 2 things. So first, we're trying to facilitate interaction sooner. So we're seeing if we see facilitated interaction sooner. Because of the very tight spacing, these trees should be interacting at a much quicker time frame than something at like 1.5 meters spacing. So that's kind of the first question is, are we seeing interactions at tighter densities sooner? And then the second one is, are we seeing a change in root prevalence and like one of our different metrics or lengths with size amounts, biomass, change based on species mixture and spacing. So primarily species mixture. Are monocultures doing different things than mixes? Because as we know, trees will diversify and niche differentiate below or above ground. So hopefully that same trend is happening below ground.
Thank you. The cartoon you had, the tube was very with an angle, but you want to have result. You're not going to go deep as much. How deep your tube are going to be?
I think 48 centimeters. Yeah. So a total depth of 42.5 centimeters will be the furthest down we would see.
Last long. The tube will last longer than your PhD.
Oh yes,
Okay.
It's very cool, this study, because we're actually going to so this is replicated at 25 centimeters, 50 centimeters, and we're going to install a rep at 1.5 meters too. So hopefully somebody will be looking at this data come 10 years time when these trees are hip
Right.
or shoulder height to see what the differences are too.
Okay, good. Thank you.
Thank you.
Any other questions? Olivier.
Thanks. So you talked about facilitation. What kind of facilitation are you expecting? In which mixtures are you expecting more facilitation, if there is, and how do you see all of that happening?
Interesting question. So let me step back. Facilitation, under the sense, it's more expression of different characteristics is more what we're hoping to see. So in the boreal forest, I think it's, don't quote me on the exact number, but it's about 85% of roots occur within the top 30 centimeters of the soil, right? So what we're hoping to see is, does that change as you mix with these mixtures, right? Because something like lodgepole pine does have a taproot, right? So it should theoretically go down first, instead of spreading out when you mix it with aspen. But when I have a plot of just lodgepole pine, are they all going to go down? Are they all going to go vertical? Especially when you're forcing interaction. Maybe that's a better word, is you're forcing interaction instead of facilitating interaction. Does that kind of answer your question, or? Okay.
Any other questions? I think with that, we'll thank you, Kyle.
You.
Appreciate it.